Kia ora koutou. Thank you very much for having me here today. And it's really been interesting hearing from all the speakers. So I'm kind of talking more from a sort of basic science background, and I'll try and explain it as well as I can. So usually during disease outbreaks, we're trying to either spot and stop them really quickly before they spread, or in some unusual circumstances, we're trying to ignite their spread. But from both instances, we have very little consideration as to understanding the ecology of the viruses and their hosts, and try to understand how they're evolving and how their evolution might the information we can gain from their evolution might help us better fight the situation. And our knowledge of virology or virus diversity is hugely biased. We know a lot about viruses that infect us and the plants and animals that we care about or ecologically important, but we have very, very little knowledge of the virus diversity that is in nature and across the tree of life. So my research tries to better understand the diversity and evolution and origins of viruses in hosts across the tree of life. So not too much, although I probably will speak about mammalian hosts, but right across this tree of life, because we're all related to all these vertebrates and invertebrates. And if we can understand viruses that have infected those, we might better understand the diversity of viruses that infect us and cause disease in us. And we can do this using a technique called metagenomics. So if we understand that genetics is the study of a gene and genomics is the study of genomes, then metagenomics is the study of all of the genomes in a sample. And so we can use this to do virus hunting. So we can do metagenomic sequencing and unbiasedly reveal all of the viruses and other potential microbial organisms in a sample. So this is a sort of technique that we can do now instead of using traditional methods such as virus culture and PCR to detect novel viruses. And New Zealand is really an interesting place to hunt for viruses because we were once connected to the supercontinent Gondwana and separated about 80 million years ago. So that means that we have lots of plants and animals that are only found in New Zealand. And so their evolution of viruses have this very unique setting. And we have things that are known to carry lots of viruses elsewhere. For example, we have two bat species in New Zealand, the lesser short-tailed bat, which is the less bat-like bat you've ever come across that usually just hangs out on the ground, and the long-tailed bat species, which is more related to species found elsewhere in the world. So these come from two different families of bats. They're both likely have come from Australia originally, however, very different ecological and evolutionary timeframes. So, for example, the short-tailed bat is the only member of this family, and it probably came over from Australia about 35 million years ago, whereas the long-tailed bat as part of a family that's found all over the world and probably arrived about 1 to 2 million years ago. So very different ecological and behavioral settings for these two bats. And so we were able to work with the Department of Conservation and see whether or not these bats were carrying lots of viruses. They do carry lots of viruses, and they also share lots of viruses, which is interesting. In particular, one that you might be interested in is a coronavirus. Bats do have lots of coronaviruses, even in New Zealand. And they even have the same species of coronavirus, which means that this virus has probably jumped from and between these bat species quite relatively recently, considering the lack of diversity between the virus that's shared between the both species. And in true New Zealand style, we've got a really weird coronavirus. Some of the coronaviruses look like other coronaviruses. This is a genome of a coronavirus. It looks very standard, and we've called it pick of a coronavirus. However, we've got another coronavirus, which is just missing this huge part of its genome. So it's got a major deletion. It's still a replicating virus, but it's the shortest known coronavirus that exists yet. So everyone's on the hunt to find the longest coronavirus, but now we've found the shortest coronavirus in New Zealand. But so these viruses jump hosts, and so it's important to understand how viruses evolve, and host jumping seems to be a sort of favorite way for viruses to evolve. And we can quantify this by understanding the evolution of the viruses. So this is like a family tree. It's called the phylogenetic tree of how related viruses are to each other. And we can understand the viruses' evolution and the host from which they were isolated. And if the evolutionary history of these viruses perfectly match like this, then the viruses have just co-evolved with their hosts. However, if they don't match like this, then we can understand that across the evolutionary time of the hosts and the viruses, they have jumped hosts a lot. And so we can see whether or not this pattern exists across virus families. So there's just a selection of virus families, and you can see lots of tangled webs, which means that viruses love to jump hosts. They actually prefer that than evolving with their hosts. So host jumping is really common. And what does that mean? Well, for example, let's look at coronaviruses again. So this is a phylogenetic tree or a family tree of the diversity of coronaviruses that we knew about in about 2020, before everyone started being a coronavirus expert. And so this was the diversity. They basically infected mammalian hosts in red and avian hosts in green, with one reptilian host noted in dark green. That was all we knew about 4 genera. However, I was involved in a disease investigation in Kanakana or lamprey. This is lamprey reddening syndrome that it causes mass die-offs in lamprey, and we got some specimens of diseased lamprey from MPI because MPI weren't able to find a potentially causative pathogen for this virus for this disease, and we found a really abundant coronavirus in lamprey. And in fact, once we found this coronavirus in lamprey, we used this genetic sequence as sort of bait to mine lots of published transcriptome data or genetic data to find lots of viruses in coronaviridae that infected both fish, amphibians, and even basal fish like lamprey. And so again and again, we can see this pattern where we find viruses that we thought had a very limited host range actually infect right across the tree of life. And so if you know anything about the evolution of vertebrates, you can see that lamprey are jawless fish, which usually kind of sit basal to other fish. However, this virus is an in-group here, meaning that there's probably this virus has jumped into lamprey at some point. Of course, 2 things: we don't know this is the causative agent of the disease. This is really hard to prove. And secondly, we don't know when this virus jumped hosts. And this long branch just means that we probably haven't sampled anything closely related to this virus, mainly because there's not a lot of this type of work done in New Zealand yet. So we don't know when this virus jumped into lamprey, and it could have been millions of years ago, of course. But viruses in aquatic systems are really interesting to study. And because lots of fish tend to have distinct host ecologies and behaviors, such as you get solitary fish and you get fish that hang out in really dense schools. So understanding the drivers of the virus diversity that you find in these organisms is really interesting. So I've done quite a lot of work across the aquatic virusphere, I guess. And again, we find the same sorts of pattern as we do in coronaviruses. Every virus family that we thought only really infected mammals and birds actually infect animals right across this tree of life. So if we look at sort of lower vertebrates, if you like, we can find every single virus that we find in us in fish as well. So of course, these are very, very distinct virus species, and there's been millions of years of evolution in most instances, and we don't have any, there's no risk for us of viruses jumping from fish to humans because of the evolutionary barriers that are just too great between those species. But it's interesting, nevertheless. So, for example, we can even go to the fish market and sample fish that are seemingly completely healthy. And I know that because I ate them. So, and it's a good way to do some field work. So you can just go to the fish market. We can sample fish, and we can reveal that there are hundreds of completely novel viruses that we had no idea about in perfectly healthy fish. And we can try and better understand what's driving this diversity in these fish. So, for example, is it their host ecology and their behavior, the way that they swim, or their sort of trophic level in the food chain? In this instances, we found we sampled lots of different lots of different fish hosts, and found that generally fish among the sort of taxonomic order had the same sorts of diversity of viruses than their more distantly related fish hosts. So fish, in fish anyway, the phylogenetic history of the host is really important for driving that virus diversity, even though we know that viruses love to jump hosts. And so I thought I should go out and do some actual field work rather than go to the fish market. So I had a PhD student involved in a sampling trip in Chatham Islands, and Chatham Islands is a really interesting place to better understand sort of fish host ecology and how that relates to virus evolution, because it's really remote. It's free of introduced species, and we sampled about 16 or so different water bodies that were completely isolated with different sort of influences of marine and salt water as well. And so a really common species is smelts across all of these water bodies, as well as other species as well, to try and understand the ecological, environmental aspects that may be influencing virus diversity in these species. We found lots and lots of viruses, and don't have time to talk about them all, but one really interesting virus, and this is just to highlight how little we know about the virus sphere, is that, so there's this is an order called Bunyaviriales, lots and lots of viruses in there. Each branch, basically, its balloon here is a virus family. There's this new virus family called Tosaviridae here. Before we sampled here, there was one virus species known in this, a complete new virus family that was discovered last year, and that was in a diseased turtle in Florida. And so the second virus that we know in this whole virus family is found in a tuna or eels, both in short fin and long fin eels in the Chatham Islands in New Zealand. And this long branch here suggests that there's lots of diversity that we just have no idea about because we're not looking. And I should say that these are very healthy eels, so they didn't have disease. And so I worked a lot in the University of Sydney before I moved here, and we did lots of fish work around New South Wales and other water bodies, because on the one hand, we have sort of similar to New Zealand, we have very sort of endangered endemic fish species, and they're coupled with, they're sort of rifling, I guess, with the problem of introduced species as well, such as carp. So carp are a really big problem in the Murray-Darling Basin, for example. The Murray-Darling Basin is a huge water system that spans about 5 states and territories, and carp account for about 90% of the biomass in these water systems. And the New South Wales government, as well as the federal government there at the time, were interested in using carp herpes virus as a way of a biocontrol against carp. And I know Department of Conservation have asked me about it as well. So there is lots of experiments done in terms of whether or not carp herpes virus might jump to native species, but these were all done in a laboratory setting, and we were interested in whether or not these fish share viruses with the native species, regardless whether or not it's carp herpes virus or other viruses, and what's the sort of relationship between their virus diversity in nature. And so we had a master's student go out and sample lots of carp around the Murray-Darling Basin. Actually, it was kind of trying to avoid bushfires at the time, but we sampled lots of invasive hosts as well as lots of native hosts. And we found very little virus sharing between invasive hosts as well and native hosts. So, and there's a few virus families that are shared, but on the whole, there's very different viruses that infect native fish and invasive fish. And this is probably because the same reason why we don't get viruses from fish is that there's too much of an evolutionary boundary to cross that species barrier, right? So the viruses have got to be able to infect your host cells, and there's just too different. They're too different to infect those hosts. I also had lots of fun in Sydney, where we built drones and flew through whale snot just to prove that we could collect viruses that are sneezed from whales. And this is kind of a way to just show that we can you can use innovation to enable exploration of really inaccessible habitats using such technology as well. And from then, you know, you can also explore viruses and things like urban invaders such as foxes. So foxes are a huge problem in Australia and in New South Wales and Sydney anyway. They're really fat and fluffy and cute in urban areas because they're really well fed because they basically eat your rubbish. And so, but in the bush though, they're really skinny and scrawny and probably a much fitter animals. So understanding the differences between the ones that hang out in urban areas versus the bush is really quite interesting. And so they had lots of different viruses in there, lots of novel viruses, and the urban ones, lots of diversity in the urban ones compared to rural ones, and as well as lots of diversity in the ectoparasites as well. And we can understand what they're eating by looking at the viruses that they have, because, for example, we found rabbit hemorrhaging disease virus, or IHDV, in foxes. So it's probably not infecting the foxes, but it's infecting the things that they're eating, like rabbits. And again, looking across this tree of life, we can just explore things because it's interesting. For example, really ancient host lineages like the Tuatara. In this naturally occurring population on offshore islands, there's about 300,000 Tuatara on this island now. And so this population density gives lots of opportunity for virus transmission and interesting virus dynamics. And so we found lots and lots of viruses in these hosts, as well as you know, again, these really long branches coming off these phylogenetic trees suggest that we just don't know anything about the diversity that surrounds these viruses. And again, across the plant kingdom, you know, even just mining already published data across these plant kingdoms. So I had a student that mined over 1,000 plant transcriptome data sets that were already published, that were being used for other things, but we could mine those data sets just to look for viruses. And we found hundreds of novel viruses and the first known viruses in lower plants. So everywhere we look, we find new viruses. And again, he quantified how these viruses were evolving, and host jumping, even among plants, was the most common way for viruses to evolve and speciate in new hosts. So why are we doing all of this and trying to understand virus diversity, and especially in New Zealand wildlife, is because I guess at the moment, there is a huge threat to our native birds, especially with highly pathogenic avian influenza virus sweeping the world, killing millions of bird species with multiple spillovers to mammalian hosts as well. So this is a real threat, and it's getting closer to New Zealand. And so we need to better understand, you know, how to first detect novel viruses, but also deal with the spread and evolution of known viruses. And so one of the bird species that are critically endangered is yellow-eyed penguins, or hoiho in New Zealand, and these hang out just around Otago on the mainland and other offshore islands, especially in sub-Antarctica. But the ones on Otago are facing a myriad of diseases. So I've been working with the Department of Conservation to try and understand what is the cause of these diseases and how we might be able to better treat and prevent them. So one disease is called neonatal respiratory distress syndrome. It seems to affect, here's some baby chicks. I work with the wildlife hospital just down the road, and they basically take in nearly 100% of chicks now because they need to better manage this disease. So it's a hemorrhagic disease, and it basically is about 100% fatal in the ones that show clinical signs, and it kills chicks within a week of life. It's been noted since about 2019, but they have no idea what was causing it. So we had we used metagenomics to try and identify a potential cause, and found a really abundant virus called gyrovirus in these tissues. So if you know anything about gyroviruses, it's probably this chicken anemia virus, which this is the gyrovirus is a genus of virus, and the chicken anemia virus is a closely related virus that that causes disease in poultry, and poultry industry vaccinate against this virus very commonly, especially in New Zealand as well. And so this is a related virus, and I mean, I say related, it's still very far apart, and it's been a long time since they shared a common ancestor, but there's lots of genetic diversity among this virus in New Zealand, among these hoiho. And what we're trying to do now is try and understand, because we think that it's likely that this virus isn't always causing disease, but try and understand the pathogenesis of this disease. And we're looking at now potentially using the chicken anemia virus vaccine to repurpose into hoiho to see if whether or not that's effective. Another disease is this diphtheric disease in yellow-eyed penguins that's been going on for about 20 years. It causes plaques to form in their mouth, and basically chicks usually die of starvation because they can't close their mouth or eat properly. For 20 years, this was a sort of unknown cause, but we looked at lots of bacteria and eukaryotic microorganisms that could be associated with chicks either having disease or being pre-symptomatic or recovered against sort of healthy adult controls. We couldn't find any differences between these microorganisms, but we did find a very abundant virus, which is in the Picornaviridae family. So picornaviruses cause things like foot and mouth disease and hand, foot and mouth disease, and lots of things that cause lesions in the mouth. So we thought this could be a potential cause. This was really abundant, and in other viruses that are quite closely related to this virus, it also causes beak diseases in avian hosts. So this could potentially be the cause of this disease. However, proving causation is a major challenge. Working with wildlife, especially endangered wildlife, you can't just do sort of challenge studies to find the potential cause. So this is really hard, but you know, having the sort of list of suspects gives us a potential way forward. Last slide, I promise. So why is it really hard to prove causation just using molecular techniques? Is because of this biased view of virology and viruses that cause disease. And that's to do with our bias sampling, right? So for example, when we look at prior to the use of sort of metagenomics, which took this arbitrary year of 2008, we thought the vast majority of viruses cause disease. And then we take the year 2020, when a lot of metagenomic studies have revealed lots of new viruses. In fact, hardly any viruses cause disease, and we need to know more about the diversity to better understand which ones are causing disease and why are they causing disease. So we need to do a lot more sampling. So we need to do a lot more sampling and include species that aren't necessarily economically important to us to better understand where these viruses might have come from and where and why they lead to disease in some hosts and not others. And look, thanks to all the people I've collaborated with, my students and staff, and all the funding and the microbiology department. Thank you.